So my name is Linda Marban. I'm the CEO of Capricor. I wanted to thank Parent Project of Muscular Dystrophy for having us here, and I also wanted to thank Pat Furlong for being the force behind getting all of you in this room. I think it's astounding that the energy of one person can derive this much in terms of advocacy to move forward treatments for an orphan disease, an ultra-orphan disease. And I applaud every single one of you for the work that you're doing for your community and for your sons, because I think this is how we actually can ultimately develop cures for disease. And I want to tell you that the only other advocacy community that I am aware of that has been this active was for HIV and AIDS. And what I heard recently is that that previously fatal disorder disease has now led to the normalization of the lifespan of those patients with triple therapy. So here's to the next few years of all of us putting our nose to the ground and developing cures and therapies for Duchenne so that these boys and young men can live out their lives as normally as possible. We are a publicly traded company, so this is a standard forward-looking statement that I have to show at the beginning of every talk. So we're a little bit of a different kind of therapeutic, and I want to help you understand why our cell therapy may be different than some of the other quote-unquote stem cell therapies that you hear about in the media and in your community. Our cells function primarily as a local drug delivery system, as I fondly like to call them. What that means is that they are going in and they are releasing a payload of these tiny little bubbles called exosomes that are filled with RNAs and proteins that actually communicate to cells to change their behavior. Our cells release each cell releases about 10,000 per day of these in culture. We don't know exactly how it works inside the body of a person because we can't image it directly, but that's sort of to give you an idea of when we inject, for instance, 75 million cells, each of those cells releasing thousands of particles per day. They're releasing a lot of potential bioactive particles into the body. And those particles, those exosomes, have been shown to reduce inflammation and encourage healing and new muscle formation. So our cells die and go away within about 4 weeks as best as we can determine. And what they leave behind is their messages of healing in these exosomes. And that's sort of summarized here in language that says that they're anti-inflammatory, anti-fibrotic, they prevent scarring, they prevent cells from dying, so cells that might be on the path to death, they help to restore them, and they stimulate blood vessel growth to feed the new muscle. The first clinical trial that we did is called the HOPE Duchenne trial. We just reported out the 6-month data in the last couple of months, where we've shown that in the boys and young men that got cells, so there's 25 that enrolled into the study at 3 different centers, Cedars, Cincinnati Children's, and the University of Florida. 13 of the boys and young men got cells, and 12 were followed for usual care. And then we followed them up both in terms of their heart dysfunction and improvement at 6 and 12 months, and then we looked at skeletal muscle as well as some other parameters at 6 weeks, 3 months, 6 months, and 12 months. And I'll be showing you a little bit of that data today. So, as I mentioned, we completed the 6-month interim analysis and reported that out. We also did a webinar with PPMD, so feel free to go back and listen to that for more detail than I might be able to provide you in this short talk today. But what I think I'd like to highlight for you this morning is that because we were originally targeting the cardiac dysfunction, so boys and young men that had a significant amount of scar in their heart and therefore were at risk for the development of heart failure, we ended up having a very high number of patients that were non-ambulant and older. And I have to say that I'm really excited that we're providing an opportunity for these older patients to have an entry into a clinical trial, because as I think you've seen, a lot of the trials are gearing towards the younger kids. And so this is an opportunity for those that are older and also those that are non-ambulant to have an opportunity to try out a new therapeutic. They had relatively large scars in their heart, but they had normal ejection fractions. So Duchenne muscular dystrophy cardiomyopathy is a little bit unusual when you compare it to the type of heart disease that an adult would get after like a big heart attack or some other type of insult. The hearts of the Duchenne patients are basically, they become hard and non-contractile because they become almost like a box. And so their ejection fractions and their volumes, the typical measures of cardiac function, remain normal almost to the end, which is why the cardiomyopathy was sort of late to the game in terms of its effects on Duchenne patients, because it was harder for physicians to figure out that they had cardiac dysfunction. Now with MRI, we're able to understand the significance of the cardiomyopathy in Duchenne. And I had the opportunity actually last night at the poster session to see the work done by a Duchenne man himself, which was extraordinary, detailing the issues that are concerns of Duchenne and Becker patients. And what I noted is that in both sets of patients, their heart disease was literally number one on the list, which I thought was really interesting and relatively reflective of the importance of new therapies for heart disease. So the therapy was safe, so we delivered the cells in this first trial in the coronary vessel. So the boys and young men in HOPE came into the cath lab, and they had a catheter put into the blood vessels of their heart. It sounds kind of creepy, but actually it's done tons of times every day in adults with heart disease and is very safe and a relatively easy procedure. But these boys and young men paved the way for an understanding that we don't have to deliver the cells by intracoronary administration anymore. The next trial that we're going to be doing we'll be able to do just by straight IV delivery, so they won't have to go into the cath lab but just into an infusion center for a relatively easy delivery. Having said that, we really didn't have any troubles. They were great, and one of the patients, I actually got to watch him, was asking if he could Snapchat while he was on the table. So I'm pretty sure he was engaged and excited. We did let him do that, but or the physicians didn't let him do that, but he wasn't impacted in a way that didn't want him to communicate with his buddies. So let me share with you a little bit of the data. This is exciting data because what we went into the study hoping was to be able to stabilize the disease. We looked at a very short time frame. The 12-month data will be out in the fall, and we hope to present that at a major meeting in the fall, so stay tuned for that. But this is a snippet of time in the life of a person with Duchenne muscular dystrophy. It could be 150th, 175th of their lifetime. So when you think about that it takes their whole life for this disease to develop, the fact that we saw a reduction in the amount of scar in a very real way in a very small group of patients suggests the bioactivity of the cells and their regarded exosomes. So of course, it didn't meet statistical significance. It was a very small clinical trial, but I think anybody can see with the red being the treated patients and the blue being the control patients that we had a remarkably visible effect on cardiac scar, and this was done in a blinded fashion, of course. Perhaps more important than the reduction in scar, because the scar in Duchenne hearts in MRI is actually pretty hard to quantify. The disease is very ubiquitous. It's all over the heart. So actually measuring the scar requires a lot of training and skill in looking at these kinds of hearts. But what we thought that was even more important and actually was statistically significant was this improvement in wall thickening. So you're probably scratching your head and saying, what is wall thickening? So wall thickening is something that you can measure by MRI or by echocardiography, which basically analyzes the amount of squeeze that the heart can do while it is ejecting blood into the body. So the stronger the wall thickening, the better you have of getting the blood out into the body and getting it to your toes and to your brain and all the places that blood needs to go. Why is this particularly important measure in Duchenne patients? You might have heard me say this if you've listened to any of my talks. It's because, as I mentioned, the hearts in Duchenne muscular dystrophy get disease in a way that's very different than adults. And what happens is when the heart usually contracts, and I know in the back you probably can't see me, but I'll just describe it. It's almost like a wringing of a towel. So there's upward squeeze and axial squeeze. And what happens is, because the hearts get hard from scar in Duchenne, is they really lose a lot of that upward squeeze, that ringing of the squeeze. And so the hearts and the patients become reliant on the actual horizontal squeeze of the heart, the axial squeeze, in order to get blood into the body. So what this data shows is that we actually potentially are getting more blood circulating around in a healthy way for these boys and young men that were in the trial. Obviously, again, red are the treated patients and blue are the controls. Going up in this situation is an improvement. So you can see clearly that the cells seem to be mediating a relatively interesting and important biologic effect. So I don't have any preclinical or mouse data to show you here today, but there's a large body of preclinical data that went into the development of this clinical trial. And I will tell you that many of the people in this room who are thought leaders in this field, as well as people in PPMD, really helped us analyze this first set of mouse data that we collected. And one of the things that we saw an improvement in skeletal muscle performance in the mice. And that was mediated by their ability to run farther and longer on a treadmill. And then when we actually went down and looked at the actual muscles, we were able to put muscles from the mice in a bath, treat it like it's in the body, and then we could measure its contraction. And we saw that they were contracting better when the mice had gotten the cells. So when we designed HOPE, what we decided to do was to do a couple things. One was to look at the cardiac effect. That's what we were gunning for. But then we also decided to look at the skeletal muscle performance. And we built in a variety of measures, one being the 6-minute walk test, and the other being the performance of the upper limb, which many of you are aware of has been developed essentially by the Duchenne community in order to look at skeletal muscle performance in patients with Duchenne who have lost or are on their way to losing ambulation. And it's a relatively clever measure, because while these tasks up here seem like the kind of thing that might not mean much to a patient, for instance, lifting a heavy can might be that patient could actually drink a soda or a cup of coffee or a beer by himself instead of having somebody help him to do it. So these are real tasks that are based on real activities that the patients want to be able to do. We built this in as a measure because we knew when we were going after large amounts of cardiac scar that we were going to have older patients, and therefore many more would be non-ambulant. And thank goodness we did that, because as I mentioned, a large proportion of our patients, in fact 80% of the treated patients, were non-ambulant. So this became a very important indicator of the functional ability of the patients in the HOPE trial. What was also interesting, and we learned a lot about the measurement of the performance of the upper limb along the way, is that soon after ambulation is lost, it appears that the shoulder level, which the entry criteria is being able to lift your arms above your head, is lost as well. So there's a short window of time where that is something that the patients retain. They lose it soon after that. And so only a few of our patients could actually perform in the upper level of shoulder level in terms of the performance of the upper limb. So I'm going to focus your attention on data that I'm going to show you in a minute on the middle and the distal level, which is essentially your elbow and your wrist and hand for those patients and people that have not had to do this yet. I think most of your kids probably have, where they're focusing on those levels of muscular performance and retention. So this is the data that we got, and let me remind you that the people coming into this trial didn't know that the cells might impact their skeletal muscle. They were coming in to get a treatment for their heart disease. So when we got this data, it really was remarkable to us, the level of improvement that we saw in the performance of the upper limb. Shown here is a responder analysis. Again, the red are the treated patients. The blue are the usual care patients. We set a parameter as a responder of a patient that had an improvement of 10%. And we did this actually really rather arbitrarily, in the sense that we're still learning about the performance of the upper limb and how to quantify it. And we thought, okay, well, what would be something that we could consider to separate out somebody who is just having a great day versus somebody that had an actual improvement. And the data gave us multiple gifts, which we are very grateful for in designing the new trial that we are about to begin in this fall. One, it showed us that the skeletal muscle performance was improved in significant numbers of patients that got the cells compared to the usual care. Two, it recapitulated almost identically the preclinical data, the data that we collected from the mice. So we think that for this particular type of therapeutic, the mice are a good reporter of what should be happening in people. And three, and thank you very much, cells, for doing this for us, it showed us where the effect of the cells seemed to be dropping off somewhere between 3 and 6 months. And I think that's evident even probably to the children in the room as the red bar gets lower to meet the blue bar. So we know now that we need to redose every 3 months, which is great. We now have been able to design the next study where we know that the patients will come in for an IV infusion, nice and easy, every 3 months. And we hope that your sons and your friends and family will enroll in the study so that we can get it done and get this therapeutic moving forward as a potential treatment for this disorder. And then finally, this data that I wanted to show you shows that there is a relationship between those that had an improvement in wall thickening, that parameter of heart function, and those that had an improvement in the performance of the upper limb, and that's the middle level, so this elbow level, which is, as I mentioned in our patients anyway, the most important parameter. And you can see that actually it's statistically significant. So this data is really important because we wanted to make sure that the patients that were seeing the effect on the heart were also seeing the effect in skeletal muscle and vice versa, that it wasn't just some happenstance kind of thing. So I think this data is reassuring to me, and I hope to you, that the cells are actually mediating a beneficial effect in the patients that got the cells compared to those that were followed for usual care. So just to sort of give you a wrap up, and I think I have spoken of this a little bit, so I'll make this relatively quick. We saw effects in both cardiac and skeletal muscle. The exploratory efficacy analysis signaled that there is a benefit of CAP-1002, our cells, in these boys and young men with Duchenne muscular dystrophy. It was safe and well tolerated, although I think that it's going to be even easier for patients when they go in to get the IV infusion as opposed to the cath lab, which, as I said, was fine, but why lay down in a hospital bed if you don't have to? And that we have to do repeat dosing to hopefully sustain and synergize the effect. So I'm just going to talk a little bit for why I think the cells are so effective in encouraging skeletal muscle performance. And I think it's largely that it's an anti-inflammatory effect followed by, on top of that, this regenerative effect. It encourages muscles to repair themselves, as well as encourage the development of new blood vessels and other good biologic activities so that then they have better muscle contraction. I will also say that all the patients in the study were on steroids for at least 6 months prior to, so the effect that we saw was in addition to steroids. What we're going to be doing in the next trial, we call it very imaginatively HOPE-2, is that we're going to target this same basic population, those patients that are older, although we won't be looking for inclusion for cardiac scar anymore. We won't be looking specifically for that. We'll be looking for skeletal muscle inclusion criteria as well as ambulatory ability. We can treat any genotype. As far as we know, we don't seem to be preferential in ability to treat any of the different types of deletions or other types of nonsense mutations, point mutations, whatever your disorder suggests. It's going to be systemic intravenous delivery, which allows us to not only treat you more often every 3 months, but also we're going to do a higher dose of cells. Since we saw such good results with this study, we'd like to see if we can get even better results with higher doses. And we'll be targeting skeletal and cardiac muscle function. My team is here, but we're also available either at our website or through Brian, who is at the booth. We'll be at the poster session. We encourage all of you to come learn about how this type of cell therapy is potentially beneficial in Duchenne. And I want to thank you for your time and attention this morning. I hope I didn't throw too much science and medicine at you, and we're always available for questions. And thank you again for being here today.